Hello students, welcome back to the channel. In this video, we are going to study about natural killer cells. Now see these natural killer cells, they are granulated cells and they target both abnormal cell as well as virus infected cell. They will not engulf the cell. Okay, they will not do phagocytosis. The damaged cell or abnormal cell or virus infected cell, they will not do phagocytosis. But what they will do, they will release chemical so that the cell death is there and they prevent the spread of the disease. How will they achieve it? With the help of two types of receptors. One is killer inhibitory receptor, which will inhibit the killing. Another is killing activating receptor, which will activate the process of the cell death. So two types of receptors are there. One is inhibitor receptor, another is killer activator receptor. Killer inhibitor receptor will try to examine MHC1. There are two types of major histocompatibility complex, that is complex 1 and complex 2. So they are trying to examine this one. Now, if this is present, it means cell death is not there. Cell can escape the death. But if MHC1 is missing, then obviously cell is going for programmed cell death. Now, presence of MHC1 marker can be changed because of many reasons. It can be absent due to the presence of viral infection, and they are destroyed or they are suppressed, the production of MHC1 is going on. So if suppose this receptor, killer inhibitory receptor, identifies sufficient number of MHC1, then obviously cell death is prevented. But if suppose they are finding that there is no MHC1 is missing, then obviously activator receptor is activated. Okay, this is activated, which is going to now kill the cell. See, so this is the inhibitory receptor. If it is able to bind with MHC1, it means cell is normal and it will not kill the cell. But if suppose it is activating molecule is able to bind here, why it is able to bind? Because MHC1 is not present. It means the response is what? That there will kill the cell. Let me explain you with another diagram. Try to understand. See, this is your natural killer cell. This is the inhibitory receptor. Please focus. This is inhibitory receptor and this is activating receptor. This is a target cell. Okay, this is a target cell. So first is ki natural killer cell will interact with the target cell. Okay, it is interacting with the target cell. Now, if suppose activation receptor will bind to the stress molecule, you can see here labeling, this is stress molecule. So first of all, this is activating receptor will bind to the stress molecule. If now inhibitory receptor is able to bind with MHC1, see here this is MHC1. If inhibitory is able to bind with MHC1, means cell is allowed to survive. Now, see the next process. This is again activating receptor will bind to the stress molecule and inhibiting receptor is not able to bind anywhere, then what will happen? It will lead to the cell death because there is no MHC1 marker. So cell death is there. So I hope so in this short video, you have understood natural killer cell. They are type of a cell which will not do phagocytosis. They will directly kill the cell. Which cell? Abnormal cell. Abnormal or infected cell. Abnormal cell means cancerous cell, infected cell, virus infected cell. And they will search only for MHC1. If MHC1 receptor is inhibitory receptor of the NK cell is able to bind with MHC1, cell death is inhibited. But if inhibitor receptor is not able to bind with MHC1, then cell death is there. And MHC1 number will decrease if the abnormality is there in the cell. Either it is virus infected or it is having as a cancerous cell. In that case, MHC1 molecule will be decreasing on the surface. So I hope so this short video might be helpful for you to learn this concept. Thanks for watching.